Okay. Yeah, I think the topic of the talk is going to be a little broader. There will be a focus on 2D crystallography, but I thought I also give a little more of an introduction for people not quite as familiar with the field. Would like you to interrupt and ask questions anytime. So this is supposed to be informal. So I'm also a adjunct professor at UC Davis since a few months. So my title has changed a little. And I'm no longer group leader at Livermore, so I'm now a senior staff scientist and PI. So let's get started. Let me see here. I actually have been in this field for working on imaging with free electron lasers for about the last 10 years, even before the first hard X-ray free electron lasers became available. I've been working with Henry Chapman, who was at Lawrence Livermore Lab, and is now the head of the CFEL in Hamburg, Center for Free Electron Laser Science, on first method development, technology development, and first proof of concept experiments for the imaging. I have two slides on Lawrence Livermore Lab because maybe not everybody is familiar with where we are. So we are located about 50 miles southeast of San Francisco. Here's a satellite image. You see the city of San Francisco, Oakland, San Jose, Silicon Valley, and Livermore is here out to the east. And then the city is about 80,000 people, and then in the northeast corner of that city is Lawrence Livermore Lab, which is about a square mile of a fenced area with a whole bunch of labs. Luckily, the LCLS, the Linac Coherent Light Source that we're using, hard X-ray FEL, is located near Stanford. That's actually not very far from us, just about an hour's drive. The Livermore Lab is one of the 17 US DOE national labs. And the main mission, together with Los Alamos and Sandia, is weapons. But in addition to that, there's a lot of other work going on basic science in many different areas. And I just wanted to point out there is always opportunities for students and postdocs to actually perform research at Livermore Lab. So what I thought I'd do is I will give a little introduction on high-resolution structure determination for biomolecule and talk about crystallography. And then talk about coherent X-ray diffraction imaging using X-ray free electron lasers, or XFELs. And then just a few words on the Linac Coherent Light Source. I think most people have seen material on that already. And then I'll go into the experimental challenges, in particular the sample delivery. That's the area I've spent a lot of time on, and present some recent results on ongoing work, also in the context of that sample delivery, but also will highlight some of the recent results in both microcrystallography and 2D protein crystallography. So why we're interested in structure? I think structure is very important to understand how biological systems work, because organisms adapt to their environments by evolution of substructures. And in the case of macromolecules like proteins, DNA, or lipids, the structure is really related to biological function. So this is a structure-function paradigm. So the hope is by better understanding structure and dynamic structure, how structures change over time, we may understand biological function. So structural biology is a science of how structures are formed and measured and how they relate to biological function. There's a whole bunch of tools that were developed to study structure. Just to name a few here, foremost X-ray crystallography, which can determine structure of molecules with atomic resolution down to 1 angstrom. The limitation is you must crystallize the material you're studying, so you basically create a regular array of the molecules whose structure you want to study. And because X-rays are energetic radiation, there's always radiation damage that kind of limits what you can do. Nuclear magnetic resonance, NMR, is another method that's being used. There's no need for crystallizations, there's no radiation damage, but the size and resolution are somewhat limited. And then there's electron microscopy, which can give either a direct photo or an electron diffraction image of an object. There's no size limit also for very high resolution images. The object needs to be relatively thin in transmission electron microscopy. And also electrons are energetic and there's radiation damage. So it's sometimes mitigated by doing these imaging measurements at low temperatures, so cryo-electron microscopy. The resolution is typically not quite as good as X-ray crystallography, but I think they're getting there with more recent developments. And there's a few others like electron tomography and mass spectrometry that I just want to mention for completeness. Often measurements with these different methods are combined to really get the whole picture of the structure and the dynamics. Structure determination by X-ray crystallography, and here's just a very general workflow diagram, very, very simplified. So you grow crystals. Let's say you want to study the structure of a protein. You prepare proteins, purify them, and then you try to crystallize them. Put these molecules in a regular array, cool them, and put them into an intense X-ray beam, either from a lab source or a synchrotron X-ray source. And then, as X-rays impinging on the crystal, different planes in that crystal lattice cause X-ray reflections or Bragg peaks that you can measure with a detector. In a typical measurement, you slowly rotate the crystal. You get Bragg peaks from different orientations. And on the upper right is a diffraction image shown from a crystal. And those Bragg spots contain the information on the structure. And then there's some algorithms that allow you to retrieve the real space structure of a molecule, which results in getting a map of electron densities. And where the electrons are dense, there's the place of the atoms, of course. So from the electron density, you know where the atoms are, and then you build some models of how the molecule looks and do some refinement and arrive at a structure. Now, of course, this is very, very simplified. For example, one of the problems is the so-called phase problem in X-ray diffraction, which comes from the fact that scattered X-ray waves are really defined by an amplitude and a phase, and you need both the amplitude and the phase for the reconstruction of the electron density. But the phase problem is that the Bragg peak intensity on your detector does not contain directly any phase information. So the intensity is really just the square of, related to the square of the amplitude, where the density is given by this equation with amplitude and phase factors in it. So there have been various approaches developed, and I don't want to go into detail of phasing. I just want to mention that there have been ways to develop to get around this problem that sometimes have to do with anomalous diffraction by heavy elements, doing multi-wavelength measurements. And it also helps when a part of the structure is already known, because you can kind of constrain your reconstruction such that the reconstruction yields the correct partially known structure, and a few other methods. Resolution really determines to what detail a structure can be resolved, and it depends really on the signal to noise of your measurement and the number of measured reflections. In practice, X-ray radiation damage limits the obtainable signal. So just to give some examples, if you have a resolution of 10 angstrom, you can resolve viral subunits. If you have about 4 angstrom resolution, you see some residues on amino acids in proteins. And if you get all the way to an angstrom and a half, you actually resolve individual atoms. So X-ray crystallography has been very, very successful. You can get structure of molecules with almost no assumptions. If you play some tricks to overcome the phase problem, there's really no size limitations in terms of the size of a molecule you can determine the structure of, as long as you can crystallize it. And you can automate the process. So at synchrotrons right now, 3rd generation light sources, they have automated algorithms and machines that can yield a large number of structures in a short time. The limitation, of course, is that X-rays are damaging. Radiation damage is an issue, which is the reason why you need a crystal to do the measurement, why you can't do it on single molecules. So you're basically spreading the radiation damage out over a large number of copies of identical molecules. And another limitation is if you do that in the crystal and the crystal is cooled, it's mostly static. So you don't see any molecular dynamics. Although that's not entirely true, there have been time-resolved studies in crystallography that were quite successful. So why X-ray field electron lasers? If X-ray crystallography is so successful, synchrotron-based crystallography has been the workhorse for structural biology. And the graph on the right, and I apologize, it's a little old data, but it shows in principle what's going on. With the advent of these 3rd generation light sources or modern synchrotrons in the 90s, the number of known structures just exploded because of the success of X-ray crystallography. So the the blue parts of these bars are the structures solved by X-ray crystallography, and then the other colors are NMR and electron microscopy. And you see the dominant number of structures is really solved by X-ray crystallography. And this plot goes to 2011. I think right now the total number of structures deposited in the protein data bank are exceeding 100,000. So big success for X-ray crystallography. Now a problem is that not all proteins actually form crystals that are suitable for X-ray crystallography at synchrotrons. In particular, membrane proteins, very interesting class of proteins, often resisting crystallization. And as of about 2 years ago, only 400 membrane protein structures were known. I'm not sure where the number stands right now. I need to look it up again. Probably about 600. And a relatively small number of these are actually from human membrane proteins. And that has to do with the fact that the membrane proteins don't crystallize very well. They are a very important class of proteins because they play major roles in cellular functions. And part of the problem with the crystallization has to do with that they're usually fully embedded within a lipid bilayer under native condition. And so the crystallization conditions for these membrane proteins are very different than for soluble proteins. So as a result, only a small number of membrane protein structures could be solved. It's getting a little better right now with the advent of lipidic cubic phase crystallization. So there's a different medium in which membrane proteins can be crystallized, and so membrane protein crystallization has made some progress and has led to new structures. But yeah, people started thinking about alternatives to crystallography for structure determination. And this is where X-ray electron lasers come in. So it was a concept that was proposed. It was actually first proposed probably mid-80s or 90s, where people were speculating what it could do if it had a very bright X-ray source. But it was discussed in more detail in the seminal paper by Neutze et al. In Nature. Janusz Haidu was the leader of that group at that time and last author on the paper. So there was the idea that if you had an X-ray source that has very, very bright X-ray pulses that are very short, so a lot of photons into a very small area in a very short time, you might be able to get a diffraction image from a single molecule. And of course, you totally destroy your molecule. So it's a totally different approach from traditional crystallography, where you try to spread the radiation damage out over a large number of molecules. Here you just hit one molecule and you totally obliterate it. But the explosion process, so they speculated, could be slow enough that if the X-ray pulse was fast enough, let's say 10 femtoseconds or 100 femtoseconds, a measurable diffraction signal could be produced from a single molecule before the molecule actually was flying apart. And they did some simulations in that paper and discussed how one could go about getting structure. So of course, one molecule would produce only a weak pattern, so you have to measure identical molecules many times, like 100,000 or 1 million times. And then there's some classification, averaging, and orientation before you can do a reconstruction. So this concept really led to the development of X-ray electron laser sources, sometimes called fourth-generation X-ray light sources. And people have speculated about free electron lasers as a source of X-ray radiation for a long time, and FELs have been around in the infrared and optical regime for quite some time. What prevented them to be extended into the X-ray regime was really mechanical capabilities of how undulators could be built. But in the 2000s, enough progress was made that the first X-ray electron lasers actually were built first in the soft X-ray regime. There was a machine in Hamburg called VOV FEL that's now called Flash. And then the LCLS and a few other sources that are being built right now. So this plot on the right just shows what a big step up in brightness this is. So the bottom axis is energy, and down here is like 1 keV, 10 keV, and around 10 keV, 12 keV is where you do want to be for crystallography. And then the peak brilliance on a logarithmic scale is basically a measurement of how many photons can you put in how small a spot in how short a time. So it's not that, and down here, these colorful curves, they're basically the modern synchrotrons we have, the brightest synchrotron sources. And there's 8 or 9 orders of magnitude in peak brilliance difference. And it's not that the FELs produce, on average, more photons than these synchrotrons. but they produce photon pulses that are much shorter, and this, because the time pulse duration is in the denominator of the peak brilliance, the peak brilliance goes up as the pulse duration goes down. So we're talking about 10 to the 12 photons in a 1 micrometer spot in some tens of femtoseconds. And we at Livermore and some other international collaborators, we actually did perform some of the first proof of concept studies, and those were done, as I mentioned, in Hamburg at Flash, at that time still called BioXFEL, where we actually did some experiments to study how fast, for the two things, can you actually get a diffraction signal from a nano object, and can you, how fast is an object, a small object, destroyed by the short X-ray pulses? And there were two papers that led by Henry Chapman, who was then here at Livermore, and there were measurements were really not done on biological samples, except some viruses that were tried by Janusz Hajdu, mimiviruses. And the results were very promising. They basically showed that the explosion happens on about 100 femtosecond time scale. So the natural pulse duration of an XFEL, which is between 10 and 100 femtoseconds, was probably enough to do the first imaging experiments. And we also showed that you can actually use the diffraction signal, the recorded diffraction image, to reconstruct the structure. So moving on, and I think most of you have heard about the LCLS, so I'll be fairly brief about that. So LCLS stands for Linac Coherent Light Source, and is the free electron laser, XFEL electron laser at SLAC, which used to be the Stanford Linear Accelerator Center, and now it's called the SLAC National Accelerator Lab, and it's also in the Bay Area, shown on this satellite picture. Is the city of San Francisco, and then the South Bay, Southwest Bay is Stanford and SLAC. They used to do a lot of particle physics experiments, and they had a 3-kilometer long linear accelerator for electrons in operation since 1966, and did particle physics research. And part of that linear accelerator was repurposed for X-ray science in 2005. So 1/3 of this 3-kilometer accelerator is now used as the electron accelerator for the XFEL. And LCLS started operations there in 2009, first in the relatively soft X-ray regime up to about 2 keV photon energy, and then since 2011 going up to 8, 9, and now 10 keV. Here's a blow-up of that site. As I said, the accelerator is here, and the last third of that linear accelerator is used as an electron Linac. The electrons are transferred into an undulator hall, and the undulator, basically a series of magnets whose poles alternate on a centimeter scale. So the electrons wiggle around in there and produce Bremsstrahlung or synchrotron radiation. And if this is done right, that radiation actually can be coherent and can be in phase for individual electrons, leading essentially to some type of lasing. And then the X-rays are further transported into a facility that's underground. There's a place called near experimental hall and far experimental hall, and altogether there's 6 experimental end stations in there, which are shown here. So the X-rays come from the left, and there's several different end stations for different experimental purposes. For the biological imaging, we started out using AMO, which was the very first end station that was built and available in 2009. But right now, a lot of the imaging experiments are going on at CXI, the Coherent X-ray imaging instrument, some at XPP, some crystallography experiments, and they're still using AMO for single particle imaging. And I should say there's another end station that's currently under construction that's also dedicated for biological imaging, which is taking up part of the XCS, X-ray correlation spectroscopy hall. So they're putting in another end station there right now, and it's almost completed. The CXI instrument itself is actually not a single instrument. It's a hutch that has a bunch of vacuum chambers in there, and there's actually 2 sample environments. The sample environment is defined by a set of X-ray mirrors that focus the X-rays down to focus. There's a 1 micrometer environment where the X-ray focus is of order 1 micrometer, and there's a 100 nanometer sample environment where the X-ray focus is of order 100 nanometers, probably more like 300 nanometers. But you focus X-rays into a smaller spot, so you get a higher X-ray fluence, X-ray intensity. And that is described in a paper. It was actually a recent paper, and when I sent the copies of the view graphs, I will add the new reference. There was just a paper coming out 2015 with an update on this instrument if you're interested in more details. Here's a picture of that hutch. And of course, the X-ray detector is an important component of such an experiment, and there's a detector array that has been developed between Cornell and SLAC called the CSPAD, Cornell-SLAC pixel array detector, that is basically a tiled detector array. You have individual detector elements, and altogether you have about 1500 by 1500 detector pixels, and the detector can be moved in the vacuum chamber closer to or further from the X-ray interaction region. So I want to talk a little bit about the practical challenges for this. So this picture that was in the Neutze paper, where you see these molecules kind of flying in like on a string, and you kind of shoot them off individually, it is a little deceiving, because if you think about the problem you have, molecules or nanocrystals or nano objects in a solution, sample solution outside the vacuum chamber, and you need to get them into the X-ray focus, which is in vacuum, inside a vacuum chamber, and you need to get them into the right spot at the right time, micrometer-sized spot. And you need to replenish those, ideally 100 times a second, because the LCLS pulse repetition rate is 120 hertz. And for some of the newer X-ray sources that are being built, like the XFEL in Hamburg, it's going to be 30,000 a second. You need to bring these particles or molecules into the X-ray beam at the right place at the right time, which is a pretty big challenge. Also, you want to run this kind of continuously without clogging, and also try to avoid wasting sample. So they have been, when we started out doing this about 10 years ago, we looked into the different options for sample introduction, and ideally what you would like to have is a kind of precision introduction of single particles, which at this point is still hypothetical. So you would bring particles in at the right time, with the right speed, the right location. So you need very precise spatial and temporal alignment and particle velocity control. And at that time, at least, it seemed like existing particle introduction manipulation methods are not quite there yet. They need to be improved by orders of magnitude. Now, people are working on this, and there's some ideas how to do this, but we're not quite there yet. So there's two other ways to do this. One is kind of a brute force approach, shotgun introduction of particles. You basically spray a whole bunch of particles. And when I say particles, I mean nanoparticles, molecules. I'm using these terms here interchangeably. Any objects you want to image. You spray them into the X-ray interaction region, just in very high concentration that statistically you'll hit a particle every now and then. And you can do this either with a particle beam that's created by an aerodynamic lens, and I have a few slides on that, or you can do it with a liquid jet, and I have some other slides on that. The problem with that approach is that you're constantly spraying, and the X-ray beam is on only a very small fraction of the time, so you're wasting a huge amount of sample. Another approach, and I think early on people were skeptical about that, was to use fixed targets, an array of particles on ultra-thin membranes that is being, and these membranes are stepped or scanned through the X-ray beam, and I have some slides on that as well. People were skeptical because anything you put in the X-ray beam is being destroyed, because the X-ray pulse is so intense. So you have to bring a fresh spot of that membrane or your substrate into the beam all the time, and that's not compatible with very high rate of data acquisition. But we have made some progress in that area, and I'll have some slides on that. It's also particularly interesting for 2D crystallography, which kind of requires that your sample is on a flat substrate. So it's still a technology that's being further developed and explored. So let me talk about the jet type approaches just a little bit. So one approach would be a nanoparticle injector that's based on what's called an aerodynamic lens. And this is technology that actually came from Livermore, from our aerosol mass spectrometry work, where you have a sample that's being aerosolized. So you bring the particles into the gas phase, and then gas goes through this device called aerodynamic lens stack, which is just a series of holes that get smaller, and the gas and the particles flow through these holes. And if you get the gas properties and the geometry right, what you do is you create a very finely focused particle beam coming out of this aerodynamic lens, and you can aim this particle beam at the X-ray interaction region. And if you're lucky, the FEL pulses that come in a train every 8 milliseconds at LCLS will occasionally hit one of these particles, and you produce a diffraction pattern. So my postdoc Mike Bogan actually helped a lot of developing this technology, and there's a whole bunch of publications on this work. So this was used for introducing viruses and larger nanoparticles into the beam already in the demonstration experiments at Flash, and it's working quite well. Also, to actually get the hit rate up is quite challenging. So the advantage of these types of injectors, the aerodynamic lens-based injectors or aerosol injectors, they can inject nearly any type of particle that can be aerosolized. It works for a wide range of particle diameters. They're pretty robust, relatively simple. There's no water background. That can be an advantage or disadvantage. So you have very little background from water and residual gas, which is an advantage for single molecule imaging if we ever get there, or imaging of small non-crystalline nanoparticles. But the issues are that the particle beam diameter is typically large, of order 100 micrometers. Also, Janet Haidu's group has made quite some progress there in focusing particles down to the particle beam down to maybe 20 or 30 micrometers now. So they have developed this technology further. It's not very easy to pulse, so the sample consumption is an issue. And that you don't have much water around the particles may actually be an issue for particles that require some water to stay in the native state, that don't like to be dried. Now, there's another injector type that's actually used, probably I would say, more frequently, in particular for nanocrystallography. And so if you want to spray a finely focused jet of water into vacuum, you can basically use a small capillary, and push water through, and out comes a jet that then breaks up into droplets. The issue is if you make the capillary smaller and smaller to decrease the diameter of the water jet, because you don't want to have too much water there for background reasons, then a small capillary tends to clog. The nozzle tends to clog when the capillary diameter gets smaller than some 50 micrometers, let's say, because if a sample contains proteins, they're sticky, or nanoparticles. And the solution to that was actually developed in a totally different context many years ago, and it was called gas dynamic virtual nozzle. What you do is you take a relatively large capillary, let's say 50 or 75 micrometer inner diameter, where you push a liquid through, and that's surrounded by a larger capillary that contains gas flow, and that gas flow squeezes the liquid to a much finer jet, down to a micrometer, let's say. And that has been put into practice by Arizona State University. They have been working on this technology for the last almost 10 years, and it has been very successful at LCLS in the nanocrystallography experiments. So Dan DuPont and Uwe Weierstahl, John Spencer's group were very instrumental in that. Bruce Dork did a lot of the development as well. So these gas liquid jet injectors, they can inject nearly any type of particle from solution. It's a very small liquid jet beam diameter, 3 micrometers. There's a possibility to pulse it. I think I hear from these guys that pulse jet is under development. The molecules are in water, so that likely preserves the native state. The water background can be an issue for some types of sample. It's not a big issue for nanocrystallography. But because of the relatively high flow speed of the water, the sample consumption is typically large. So in a typical nanocrystallography experiment, things like 100 milligrams of proteins are required, which is a huge amount of protein if it's not a very abundant protein. There's a very recent development. I mentioned the lipidic cubic phase as a medium to crystallize membrane proteins. Arizona State has adopted this liquid jet injector and produced the lipidic cubic phase jet or injector. So the LCP is much more viscous than water, so it's flowing much smaller, but the principle is kind of the same. You squeeze it out of capillary, and you kind of reduce the size bit by the gas flow. And you get a much smaller flow speed, which is great for reducing sample consumption. And as in addition to some of the recent science results that were published in Science, and I'll have a view graph on that later, there's also a Nature Communication paper by Uwe Weierstahl describing this development further. And lastly, fixed targets. This is something we actually picked up in Livermore because we were interested in 2D crystallography, but we also looked into using that for 3D microcrystallography. And what I mean with fixed targets, it's sample support, a substrate containing thin membranes that is moved in steps or continuously across the beam. I think we realized after the fact that fixed targets are actually a bad choice of word, because the targets are not totally fixed, right? They're at a fixed value, or they're solid substrates, kind of fixed while you shoot at them. But then in between shots, you move the sample, or you continually scan the sample across the X-ray focus. Just for the nomenclature, some people use LJ-SFX for the liquid jet and FT-SFX for the fixed target serial femtosecond crystallography. So I'll use FT-SFX, and it means fixed target serial femtosecond nanocrystallography. So why is it interesting while the liquid jet works so well? So one of the advantages of the fixed targets is there's a potential for a drastic reduction of sample consumption. And my former postdoc Mark Hunter actually went through some calculations, and you can improve over the liquid jet injection by many orders of magnitude. You can optimize the crystal density without affecting the flow properties of a liquid containing the crystals. So sometimes with liquid jet, you have an issue. You can't get the density up to what you really want because the jet doesn't flow at the high density anymore. There's no clogging. You can use a fixed target approach for sample screening. So if you have only small amounts of sample, you put them down on a fixed target, you know where they are, and you can do screening to see whether your crystals diffract or not. There's less background from a thin substrate, and when I say thin, I mean like a 30 nanometer membrane of silicon nitride or carbon that's kind of supported by a larger silicon wafer or some other type of wafer. The background from that is much smaller than for LCP. And there's some automated sample preparation techniques that we have been exploring, like inkjet printing, where you can actually put down exactly the right amount of sample. And then, 2D crystallography, I mentioned that, which requires your 2D crystal to be on a flat substrate, and you need to be able to tilt your substrate. It's really only possible with a fixed target approach. I'm not sure it will work very well with liquid jets. There's some practical issues with the fixed target approach, and foremost, the vacuum protection of the sample. So if you have a sample that needs to be hydrated, if you put that on a solid substrate and put it in vacuum, it will dry out very quickly. So you need to think about how to protect your sample from vacuum. The extra background from substrate, while I said it's not as bad as for LCP, it could be an issue if you have very small nanoparticles or if you want to go to single molecules. There could be additional damage, and I haven't really talked about damage much other than saying that X-rays are potentially damaging your sample. Well, it turns out for fixed targets, because you not only have your sample in the X-ray beam, you also have the thin membrane or the substrate in the X-ray beam. Fast photoelectrons produced in that substrate may cause additional damage. So the damage may actually appear faster than in let's say, a liquid jet. And then there's always the issue that with fixed targets, the data acquisition tends to be slow, in particular if you are stepping your sample through. You basically move your sample, you stop, you shoot at it, you move, stop, shoot at it. It's a relatively slow process. And there's always concern that you don't only hit your sample that's sitting on a very thin membrane, but you might hit the frame holding the thin membrane, which is something the beamline operators absolutely hate, because if you hit anything that's not thin, you produce a large scatter signal, and if you hit something that's crystalline, you might even produce very strong Bragg peaks or streaks, and that could damage your detector, just too many photons hitting one pixel at the same time. So here's an example for one of these sample holders that we used at LCLS. It's an aluminum frame that is made to hold wafers. In this case, these wafer pieces were silicon, 1 inch by 1 inch mostly. You see the bright spots, which are little individual membranes. So there's a silicon wafer that has a silicon nitride layer on it, and it's etched out from the back, resulting in membranes that are supported by the silicon. The holder can accommodate a larger number of these wafers. I think the latest one can probably do 12 of these 1 inch wafers. You can also adapt it to hold other things like TEM grids or other types of substrates. And then there's motor stages that move the that move that holder around, so you always hit a membrane where you want to hit it, and this can be automated. Currently, these scripts allow you to do 1 step and shot per second. We have also, and then here's on the right is the principle shown again. So we have a silicon wafer. It's a cross section with a thin layer of silicon nitride, and that's usually produced before you etch out the silicon from behind the silicon nitride, and remaining is the silicon nitride membrane. And you can shoot X-rays onto that either from the front or the back, and then if a sample is present, you will get diffraction that you then can measure with the detector. We've also explored wafers with thin and long windows. Instead of having little square windows, we have these elongated membranes. They're also 30 or 50 nanometers thick. They're about 1 centimeter long, 200 micrometers wide. And what you can do with a long membrane is you can have the X-ray field electron laser like LCLS pulse at a fixed rate, let's say 10 hertz. And then you can continually move that membrane through the X-ray beam, and you get about 100 shots or so into 1 centimeter, if you space them out by 100 micrometers. So this way you can speed up the data acquisition. You can also, instead of silicon nitride, you can cover these windows with ultra-thin carbon foils, produce carbon substrates. And there's no reason why this needs to be silicon. We have also explored other types of wafers or holders, including some plastic wafers. And depending on the application, you can get away with fairly simple plastic wafers. I did not talk much about the vacuum protection, but for the 3D crystallography, you can, for example, embed your microcrystals in something that is vacuum protecting, like an oil, and it has to be an oil that's approved by the beamline, so it can't be any oil. They don't like certain oils in the vacuum chamber, but Paraton N is one of the oils that we're allowed to use, and that works very well. Of course, you might also be interested in having a fixed target measurement on a sample that is fully hydrated. Our colleague James Evans, who is now at the Pacific Northwest Lab, has developed a technique that does exactly that, and it's essentially two of these wafers with thin membranes just put on top of each other with little spacers in between, and in this case they're gold spacers. So you have a bottom wafer, a silicon nitride membrane, there's some lines of gold on top of it, you put a sample down in a liquid solution, and then you take another piece, turn it around, and put it together. And what you produce is individually enclosed compartments of sample. So instead of having a 100 by 100 or 200 by 200 micrometer large single membrane, you basically have a little compartment that is that size, a square compartment or elongated compartment where the water is enclosed. These gold spacers serve as spacers, so you enclose a small amount of water. There's not a large amount of liquid in there, and they also seal individual cells from each other. So you can have a wafer that has of order 1,000 of these cells, and you can shoot out these membranes as you do with the other fixed target approach, except that now your sample is in water. So I want to turn a little bit to some of the recent results and some ongoing work. Are there any questions so far? Okay. So nano and microcrystallography, it's actually one of the first things that was tried at LCLS, and I think it confuses some people, because when we show these pictures from the Neutze paper, 2000 Nature Neutze paper, where individual molecules are flying in the beam and they hit on the fly, and XFELs were basically touted as a solution to the crystallization problem, that you could do single molecule imaging. And then we have an XFEL, and what do people do? They do crystallography, just with smaller crystals. Now, to be fair, the X-ray flux that we have available at LCLS right now is probably not quite enough for true single molecule imaging. And a lot of people feel micro and nanocrystallography is a stepping stone towards smaller and smaller objects and towards single molecule imaging. So some proteins may form nano and microcrystals, but really not many large enough crystals of sufficient quality for traditional X-ray crystallography. There's also crystallographers who speculate that nanocrystals may have better order than larger crystals. Of course, there's others who object to that because there's more surface and maybe more disordered. And I think that's still an open question. But it's nice that you're actually dealing with a micro or nano object, and nanocrystals as small as maybe 200 by 200 nanometers have been measured. But because they're crystalline, you have the Bragg reflections, you have the enhancement of the signal in the Bragg peaks rather than just a low signal noise diffraction pattern. Bragg peaks, the intensity of the Bragg peaks might also be useful to assess residual radiation damage. So just because we do diffraction before destruction doesn't mean there's no destruction at all. There is, on some time scale, there will be damage visible, and there's still a field of active research. How short does the XFEL pulse really have to be before we don't see any kind of damage anymore? And the times get shorter and shorter the closer people look. So what I mean with you can use the intensity of Bragg peaks to assess damages, if you know how large your nanocrystal is, you can calculate how intense a particular Bragg peak should be. And then as disorder sets in during the damage process, the intensity of that Bragg reflection goes down. So when you compare the measured Bragg intensity to the one that you calculate, you might get an idea how much residual damage may still be there. Nanocrystals work well with liquid jet injector because the Bragg peak signal is relatively large. You don't care too much about the water background. And the liquid jet injector, as I said, has the advantage it keeps crystals and protein samples hydrated. And it works very well with the present LCLS beam parameters, meaning the peak brightness that we have there, which is probably not quite sufficient for single molecule imaging yet. And there's methods developed in synchrotron crystallography, time resolved studies, and also the data analysis developed for traditional crystallography that both can be applied in nanocrystallography as well. So this picture is actually from one of the first experiments, and 0 femtosecond nanocrystallography at LCLS. This is a picture from a very first experiment at AMO where the detector was not the CSPAD but a different kind of CCD detector. A liquid jet was used to shoot a jet of microcrystals of photosystem 1 across the interaction point with the X-rays. The X-ray pulses were interacting with crystals, and down here is schematically shown a 4 micrometer diameter water jet with some small crystals in it. Then the X-ray beam diameter in this experiment was about 3 micrometers. And occasionally a crystal is going to be in the right place at the right time, and you'll get a diffraction pattern. Photosystem 1 was used as a first test case. It's somewhat counterintuitive. It's one of a very large protein complex, but it's one that actually forms crystals. It's a membrane protein that can't be crystallized. And so instead of using some small protein crystals, people were going for large molecules right away and just basically trying it. The structure was known from traditional crystallography, so it was a good test case. And in these first experiments, the structure could be reconstructed to about 8 angstrom resolution, which was commensurate with the X-ray photon energy that was available then, which was only 2 keV. I mentioned earlier that for traditional crystallography, you really want to be at 10 or 12 keV. But this was published in a seminal paper in Nature in 2011, together with another result that was obtained on mimiviruses. And I'm not talking much about single particle imaging today, but there were 2 seminal experiments performed right after LCLS switched on. One was this nanocrystallography experiment, and the other one was the imaging of mimiviruses. Oh, and calculated what the radiation dose was that these proteins were exposed to. It was about 3,000 megagray, which is 100 times above the damage threshold that usually limits measurements in synchrotrons. Of course, when you have larger crystals in synchrotrons, you can always move to a fresh spot in your crystal and get around the radiation damage to some extent that way. So these were the 2 papers I mentioned on protein nanocrystallography and on the imaging of single mimiviruses. So in 2011, the other end stations at LCLS became available, in particular CXI, the Coherent X-ray imaging instrument, and very quickly a lot of other results were produced. So CXI, measurements at CXI can be made at up to 10 keV photon energy. I think they're pushing that higher and higher. I think they're approaching 11 right now. And high resolution now, approaching almost atomic resolution, 0 femtosecond nanocrystallography was demonstrated using that CSPAD detector that I showed earlier. At first, it was put in model systems where the structure was known, lysozyme, and then photosystem 1 and 2. And then very quickly, also the first new science results were produced. And I have some view graphs on these things in a minute. And more recently, we also did some high-resolution time-resolved studies using time-resolved crystallography type approaches. So I'm just showing this because there's a pretty diffraction pattern from a nanocrystal of photosystem 1. And this was now done with 9 keV photon energy, and it's a single shot pattern. So this from a single crystal, 40 femtosecond long X-ray pulse, and a lot of Bragg peaks in that diffraction pattern. The ring that's in this picture is at 3 angstrom resolution. So resolution gets better, meaning smaller numbers and higher resolution as further you move out. So I think in this case, signals were observed almost out to 3 angstrom. And structure could be reconstructed to that resolution as well. And I think this was work done by Alex Bronger at Stanford. First new science result, I think this was the very first new science result in terms of structure, came from a group led by Lars Latteke from Hamburg. They were measuring nano and microcrystals of cathepsin B, which is a protein from a bug called Trypanosoma brucei, and it's the causative agent of the African sleeping sickness. And this protein is involved in the pathogenicity of that. And so they studied protein, microcrystals of that protein. Part of the structure was known beforehand, and they reproduced that structure to about 2 angstroms at LCLS, but they also found new components in that protein that were not previously known. And interestingly, these crystals were actually grown naturally inside bacteria. So they were not grown from purified protein, but the protein was expressed in bacteria and formed naturally crystals there. And people speculate that this additional peptide that's binding to the protein that was found in this structural study is an inhibitor that prevents these bacteria from actually being affected by it. So it was a very, very interesting result. And there were some other parts of the structure that were previously not known that had to do with post-translational modifications that were also observed in this measurement. It's just a picture of these cells that were infected by a modified virus that carried the gene for the protein. Here's a diffraction pattern. Again, about the same parameters, 9 keV, 40 femtosecond pulse. Now, one thing I would like to stress is for the in order to get really enough data to reconstruct the structures, you have to hit a lot of these microcrystals, because each crystal is in a random orientation and only it gives you Bragg reflections that correspond to this random orientation. So you need to have a lot of hits of crystals that are all in different orientations to probe the whole Fourier space and all the reflections that are possible. And the data set that was produced here for the system 1 was collected over, I think, over 8 hours of data collection, where almost 4 million frames or images were collected. Not every one of these images that spread out for every X-ray shot contains diffraction patterns. But out of these almost 4 million frames, 357,000 contained measurable Bragg peaks, crystal hits, and then about a third of those were actually good enough that it could be indexed and could be used for the reconstruction. So it's a large number of crystal hits that you need, and that's a very different way to take data compared to a larger crystal at a synchrotron, where you maybe measure the crystal in some tens or a few hundred orientations. So the actual number of image frames you use in synchrotron-based crystallography is much smaller than here. I did mention the phasing problem early on in my talk. So people were worried that some of these phasing approaches might not work with XFELs. But there was a paper just about a year and a half ago, or 2 years ago, by the Heidelberg group on a de novo protein crystal structure determination. What I mean with this is the reconstruction of the diffraction data does not require any de novo, any prior knowledge about the protein structure itself. And the way they did that is they labeled the protein, and I believe it was lysozyme. They labeled it with gadolinium, which is a heavy metal atom, and that leads to some anomalous effects in diffraction that can be used for phasing. So anomalous diffraction was one of the methods that can be used to overcome the phase problem, and so this was demonstrated, and pretty much checked off that de novo phasing can be done at FELs. And then people also looked into photosystem 1 and photosystem 2. Particularly photosystem 2 is very interesting. There are several different groups, 2 different groups or collaborations working on photosystem 2 at LCLS, getting first relatively high resolution diffraction patterns. So photosystem 2 is interesting that it's really the water splitting molecule in the process of photosynthesis. So photosystem 2 goes through a photocycle that involves structural changes. As it goes through the photocycle, 4 photons are absorbed, so not simultaneously, but subsequently. And it goes through the photocycle, and in that process splits water into hydrogen and oxygen. So photosystem 2 is actually responsible for for plants producing oxygen. At the core of photosystem 2 is a manganese cluster, and these manganese atoms change oxidation state in that photocycle. So in principle, people understand what's happening in photocycle, but there haven't been any high-resolution, time-resolved measurements on how exactly the structure is changing as the molecule goes through the photocycle. And with Peter Forme led some experiments that actually looked into structural changes in photosystem 2 during the photocycle. And this was done in what's called a pump probe experiment. And this is from an older experiment that was already done at AMO, but the setup was essentially the same at CXI, where you start out with crystals that are, let's say, in the dark state, and then you want to move them to the first excited state by flashing them with light. And this is done by shining light pulses onto the liquid jet as it comes out of the liquid jet injector, and give them a brief flash of light, and then a large fraction of these photosystem 2 molecules go from the dark state to the first excited state, or S2 state. And you can then probe these crystals with X-rays coming from the X-ray beam. And this is called pump probe because you pump your system that you want to study with light, and you probe it sometime later with X-rays. And of course, you can trigger the optical laser with a known time delay before the X-ray pulse. So you can adjust the time between the optical excitation and the X-ray probe, which probes the structure. And the hope is that at some point we'll be able to produce what's called molecular movies with this method. So we basically take a whole bunch of data at one fixed pump probe delay time, and then we change the pump probe delay time to a larger time, and so on and so on. So each one data set would be a single frame of a molecular movie. And that this actually can work in photosystem 2 was demonstrated by experiments that were led by Petra Forme, published in Nature 2014, where we studied the structural change as the photosystem goes from S1, the dark state, to S2. And shown here is just the core of the molecule, the manganese atoms and oxygens and calcium. And the blue lines indicate electron density in the dark state, and the yellow line indicate electron density. Excuse me, it's actually the second excited state. So this was a molecule that was excited by 2 subsequent light flashes, because the structural change going to the first excited state was presumably not as large. So we're comparing the dark state with the double excited state, so which is indicated by the electron density of the double excited state indicated by these yellow lines. So this was very, very exciting result. And now this was still at a somewhat moderate resolution of about 5 angstrom, and I think there are all kinds of issues with getting very high resolution data from photosystem 2. Also, the other group led by the Cantor-Vittal, they I think have about the same type of resolution. So there's still room for improvement. More recently, there has been work, and I don't think I have a slide on that, on a time-resolved measurement on photoactive yellow protein, P by P, that was led by Mario Schmidt from Wisconsin, experiments that I was involved in as well, where for the first time we actually saw structural changes on microsecond time scales with near atomic resolution, a resolution of about 2 angstrom. And those experiments were repeated even more recently, just a few months ago, where we were actually trying to study very, very fast structural changes that occur in the time scale of picoseconds. And that gets into a time regime that is no longer possible to be studied at synchrotrons for reasons of time structure of the X-ray pulses in synchrotrons. You can't build up enough signal on that shorter time scale. But XFELs in principle allow you to do time resolved studies down to the pulse duration of the X-ray pulse, which is 40, 50 femtoseconds. Should mention a few other things before turning to 2D crystals and wrapping up. So there have been SFX experiments on membrane proteins. I mentioned LCP as a medium in which membrane proteins can be crystallized, and LCP can also be injected with this new LCP injector. So it was experiments led by Vadim Cherezov, looking into crystallography on cheap protein coupled receptors, and there have been very few GPCR structures in the protein data bank until very recently. They're notoriously hard to crystallize by other means, and just very hard to get structures from. And he showed at LCLS that it's actually relatively straightforward. And the advantage of the LCP jet is it flows much slower than the water jet, which also means the amount of sample you need to inject to get a full data set is much smaller. So we're talking about sub milligrams, 300 micrograms of protein. And I think Vadim has since then had several more successful beam times at LCLS, and each one resulting in additional structures from GPCRs and other membrane proteins. So very, very successful effort. I want to briefly say something about the fixed target approach for 3D microcrystallography. This is something we looked into at Livermore, and at Mark Hunter in particular, my postdoc. So we had protein microcrystals on thin substrates, like the one that's shown here with elongated membranes, embedded in paratone N, which is that oil that protects the crystals from vacuum. You can put your sample on the membranes, and you have a holder, many of these wafers, and then you scan the sample through the X-ray beam. For these long membranes, we can do this at a data acquisition rate of 10 hertz. 10 hertz while you scan along a membrane, and then of course you have to realign to go to the next row, which takes a little time. But overall, the data acquisition rate for these type of experiments was much higher than what people had previously done with fixed targets. And what we could show is that this paratone N embedding works very well, in particular for this protein that we looked at, and we got Bragg peaks to 2 angstrom resolution, and we could show that the Bragg peaks were in the expected positions from what we know from the known structure of Rep24. We've since expanded this work. So the top half of this view graph kind of summarizes the measurement I just showed you on the previous view graph, and was a discussion about how much sample would actually be needed to get a full data set if the experiment was somewhat optimized with some moderate assumptions, not crazy assumptions, and we arrived at 100 micrograms that would be required, which is a factor 100 or 1,000 better than for liquid jet. And then more recently, we have done experiments with windowless high repetition rate, high repetition FT-SFX, and what it means windowless is instead of having just membranes that are carried by a silicon wafer, you could get away with the wafer altogether if you just put your sample in a thin foil, sandwich it in between 2 foils, for example, and the foil itself has a large area. Then you don't need to worry about where you shoot with the X-rays. You can just scan in a meander pattern continuously through a whole foil. And we actually did experiments where we ran LCLS at the full rate of 120 hertz with this approach. And you can optimize the crystal density, so you get a near optimal number of hits. So what we've shown there, and hopefully this will be published soon, what we have shown there is that you can approach the data acquisition rates that are possible with the liquid jet. at least for LCLS. Of course, if newer XFELs like the Hamburg XFEL come online that operate at 30,000 pulses a second, the story is going to be different again. So I want to, at the end, I want to mention a few things about 2D protein crystals. So people believe that 2D protein crystals may provide a path for X-ray imaging on membrane proteins. And I did show something on membrane proteins before, so I don't need to go into that anymore. But because membrane proteins are normally fully embedded in a lipid bilayer under normal native conditions, there's a way to reproduce that and to produce what's called 2D crystals, which is really a monolayer, an ordered monolayer of these proteins where you have lipid and the hydrophobic parts of the membrane protein are stuck in the lipid, and if you produce those right, the proteins actually form a regular array. It's like a crystal that just has one layer. Here is an electron microscopy image of a 2D crystal of ATP synthase, which is a large molecular motor. And 2D crystals have been used extensively in electron microscopy studies of membrane proteins. So it's not a new method. We're trying to do diffraction imaging on these objects in XFEL. So people have tried to do X-ray diffraction studies on 2D crystals at synchrotrons, and you usually don't get enough signal before you damage the 2D crystals, at least in transmission. I think there have been some grazing incidence measurements that were successful, but in transmission, I think people have been unsuccessful. So we were trying to do this at XFEL in transmission. The challenges are, of course, that the signal from such a 2D crystal is much weaker than from a 3D microcrystal, because your signal goes like n squared. And if you have only one layer instead of multiple layers, then you get a much weaker signal. You also need to optimize the 2D crystalline patch area. You want to really fill the whole X-ray beam diameter with crystalline material, which is a little bit of a challenge because what the methods people have developed before for electron microscopy didn't require the crystalline patches to be all that large. And you also need to, because we can't see those 2D crystals when we put them down on the substrate, we have to develop methods that allow us to cover a large fraction of the substrate with the 2D crystals. So when we shoot blindly with X-rays, when we shoot blindly at these membranes, we have a high probability of hitting a 2D crystal. And of course, the other challenge is dehydration needs to be prevented. And then in addition to the challenges that we have for 3D crystals, for 2D crystals, because they are lying down flat on the surface, you only look at one orientation. So in order to actually get diffraction information from various angles, you have to tilt the sample. Okay, so we can profit from some developments that were done for EM. There's some synergies. The sample preparation techniques can be used, but as I said, they need to be adapted because we need to provide a large area coverage and patch size. So in electron microscopy, people can use low dose electron beam to locate those crystals on a TEM grid or a membrane, and then basically move the electron beam to that good spot and do the imaging there. In our case, we can't do that with X-rays because we don't get enough signal from an attenuated beam, and looking with the full beam destroys the sample. So we have to rely on trying to get a large area coverage with 2D crystals, so even random hits on membranes have a high probability to hit a 2D crystal. So the methods of how to grow these crystals need to be adapted. In EM, often people grow the 2D crystals actually in solution, and there's some published procedures how that can be done, and then they're kind of fished out onto a TEM grid. A TEM grid is usually a very small object. They're like 3 millimeters in diameter with a bunch of openings where a thin carbon film on it, for example, where the 2D crystals get deposited. In our case, we have a square-inch wafer or something of order square, a much larger area. So these methods where you fish out 2D crystals from a solution onto a substrate, they don't work very well with these large wafers. So we're still working on improving our method to produce these 2D crystals. But yeah, in the meantime, we have gotten some interesting results from 2D crystals. So already in the first beam time, that was about 3 years ago, we were looking at 2D crystals of streptavidin, and we're looking at 2D crystals of bacterial rhodopsin. And we actually got measurable diffraction signals in transmission for both of these membrane protein to the crystals. I think bacteriorhodopsin is a membrane protein, streptavidin is not. And we got single-shot resolution in these diffraction patterns of about 8 angstrom. So we could use the data to basically at least do some sanity check showing that the diffraction images we got could reproduce to the projection images that are expected from the known structure of these molecules. And then, since then we have worked on improving the data analysis, also improving the data acquisition, reducing the background in the signal, increasing area coverage, and every beam time we had since we make steps forward. So by merging a small number of these diffraction patterns, we can improve the resolution to 7 angstroms that was shown 2 years ago. We've shown that we can tilt 2D crystal samples as necessary to acquire data for structural reconstruction. So we actually built or CXI had a tilt stage, and we could tilt samples to large angles. In principle, X-ray should allow you to see through a tilted sample up to a very large angle. In electron microscopy, you're usually limited to about 50 or 60 degrees. So that could be a potential advantage of X-rays. Shown here is only, what I'm showing here are actually averaged images that show powder patterns. So the diffraction spots get blurred out into rings here. Otherwise, you wouldn't see anything in these images. But you see that at 15-degree tilt angle, this pattern gets elongated. If it's weaker up here, and there's some elongation, and that elongation gets larger as the more you tilt, which is what is expected. We have also looked into sample preparation issues. So the original way to protect these 2D crystals from vacuum was not peritone N embedding. You couldn't do that because there would be probably too much oil on the crystal, and it might, if it's a membrane protein 2D crystal, it might also fall apart in that oil. So the way that was already developed for electron microscopy was to do some sugar embedding, essentially replacing water with sugar molecules, and the hydrogen bonds that are provided by water are now provided by the OH groups in the sugar. So it's a method that we adapted from EM. The problem with the sugar embedding is it does produce a measurable X-ray scatter background, as shown on the left, where there's a ring of signal that comes from the sugar. When we use these hydration cells, I mentioned those earlier, developed by a colleague James Evans at PNNL, where you sandwich a sample in between 2 thin membranes and enclose some water. You get away with much less sugar or no sugar at all. In that case, the sugar background is drastically reduced. Further optimization of the sample preparation and also further optimization of how to select patterns for averaging have led to further improving resolution of these patterns. Anton Barty from CFEL has worked a lot on that, and Bill Petrini. So we're down now to about 4 angstrom resolution. So every time we improve the averaging and average more pattern, we extend signal out to higher resolution, which means there is signal out there. It's just not visible in an individual diffraction shot. So we need to average multiple shots before we actually can visibly see that signal. So this is very encouraging. We also have done some studies kind of comparing experimental results with what we actually would expect from 2D crystals. So if you assume a more or less ideal 2D crystal of a protein of known structure, and both BR, bacteriorhodopsin, and streptavidin have known structures, you can calculate what the expected signal would be, making some assumptions on the incident X-ray flux that we get some numbers from the CXI team about how many photons are really in that focus. And it's a complicated plot, and I don't want to walk all the way through it, but the story is essentially that the experimental signal is smaller than expected for the ideal X-ray focus. And there's also some disagreement in structure factors, and the signal seems to fall off at higher resolution and high Q. And currently trying to investigate this a little further, and we hope to get more beam time to go into more detail there. But there's many reasons why the experimental signal could be smaller than expected and could also fall off at high resolution faster than expected. And of course, it's very hard to distinguish between the different cases here. So first of all, the 2D crystal quality could be not as good as we think. So I said we use ideal crystals as baseline for our models. So there could be imperfections, disorder, local ripples on the 2D crystal that would reduce the intensity of the Bragg peaks. That could be caused by sample preparation or by sample dehydration. Another possibility is that the 2D crystal patch size on average is smaller than the X-ray spots. I mean, we know roughly what the patch size is, but we don't have really high statistics on that yet. But if you don't use all the X-rays to produce a diffraction signal, of course your signal is smaller than what you would calculate for the ideal case. And then there's also some evidence that the X-ray focus in that 100 nanometer sample environment at CXI is not ideal. There could be a possibility that there's fewer photons in the actual focus than assumed. And the focus may not be where we assumed it to be during the measurement. It's a very tricky enterprise to hit a sample right in the focus of the 100 nanometer sample environment, because the beam is rapidly converging and rapidly diverging. So just being off 0.5 millimeter in C, downstream and upstream of the focus, makes that focal spot larger by noticeable amounts. And of course, there could be damage during the X-ray pulse that is somewhat different, or in addition to the damage that people are used to from 3D microcrystals. And it has to do with high energetic photoelectrons being produced that could go through a sample. I don't have time to go into detail, but there's all these different factors that contribute to bringing our signal down, and we're hoping to investigate this a little further. So I think I should wrap up here to leave some time for questions. I just want to briefly summarize that X-ray crystallography at synchrotrons has provided thousands of atomic resolution protein structures, has been struggling in particular with membrane proteins, which most of them don't grow nice crystals, and that the diffraction imaging at XFELs promises to overcome this crystallization problem and the radiation damage limitation problem by means of diffraction before destruction. In principle, single molecule imaging should be possible, but we are still somewhat away from this. I think XFELs are just at their beginnings. I mean, the LCLS has been operating at high X-ray energy in the high X-ray, hard X-ray energy regime only for about 3 or 4 years now. And other sources are coming online now. They have been in Japan, SACLA, and now the XFEL in Hamburg and the one in Switzerland. But even though they're fairly new, already in the last few years we have obtained high resolution, near atomic resolution protein structures. There have been unknown structures that have been solved. There have been very encouraging results from two-dimensional protein crystals of membrane proteins. And I didn't have time to really go into single particle imaging, which is even more challenging because those deals usually with non-reproducible particles. So you don't have this advantage of having the Bragg enhancement in your diffraction signal. So the diffraction signals are usually much weaker and require much more sophisticated ways of averaging and data analysis. But there have been very encouraging results in this area as well. And LCLS recently has started a single particle imaging initiative to bring researchers from many groups together to help them to solve the practical issues related to single particle imaging, from beam conditioning to sample preparation, sample injection to data analysis. And this turns out to be a very fruitful collaboration, non-competitive collaboration right now. I think true single molecule imaging still needs to be demonstrated, and it might actually require more X-ray fluence than the first generation XFELs provide. But I think there's paths that are being discussed in increasing the flux or the peak brilliance of XFELs in the near future. And to conclude, I just want to put up some acknowledgments of our team at Livermore here, and then we have had very intense collaborations with John Spence and his group, and Petra Fromme and her group, and others. And then overall, data I showed were actually from a much larger collaboration of researchers, and I probably forgot some of the names here, but it's a large group of people who has worked in this area very hard over the last 5 years. Okay, thank you, and I'm happy to answer any questions you may have.